just because we have a lot to cover today and we're super excited to get through it. So my name is Bassem Gwali. I'm accompanied by Janaka Raman, Subramanian, Silambar Samaskamani, and Ali Ukar. We are delighted to have you here with us today. We hope that this is of value to you today. We're going to cover ALK and MET exon 14 skipping mutations, so it should be pretty exciting. So these are our disclosures and our disclaimers. So today we're going to talk about the Hutchmed pipeline. So I think people know that we tend to talk about a company's pipeline with each of these. I think that that's somewhat unique. It always ties in with the cases that we talk about. So we're going to just briefly talk about Hutchmed's pipeline. We're going to do two non-small cell lung cancer cases. One is going to feature an ALK fusion, and the other one's going to feature MET exon 14 skipping. Now these are reversed, actually, they're in reverse order. And then we'll have concluding remarks. So let's talk about the Hutchmed pipeline for a moment. So the first drug is savolitinib. This is a MET inhibitor. It's actually approved in China. They're doing trials now. Hopefully they're going to get FDA approval in the US, but they've had some pretty decent data. We'll talk about it later. Surufatinib is their VEGFR 1, 2, 3, FGFR1, CSF1R inhibitor. This drug actually just recently was submitted to the FDA for neuroendocrine cancer and was rejected despite having pretty good data on face. It is approved in China, but this is an interesting drug. You can see they're testing it in neuroendocrine tumors, still in some solid tumors and the like, gastric cancer, small cell lung cancer, sarcoma. But it had a pretty bad go of it with respect to neuroendocrine cancers and with respect to the FDA of late, which is unfortunate. We're going to talk about fruquintinib for a minute. This drug is interesting, and I think this drug, you're going to hear a lot more about it. And the reason why is they recently had a phase 3 trial that showed in late stage 4 colon cancer, colorectal cancer, against best supportive care, they had a pretty phenomenal results. They had a median overall survival of 9.3 months relative to 6.6 months with respect to placebo. Median PFS was prolonged by roughly 2 months. It's actually better than Lonsurf and regorafenib, which are both approved in this late stage setting. So they're actually under review by the FDA right now. They're likely going to get approved, I would think, in late stage colorectal cancer, which will be nice. Take that with a grain of salt. The FDA has been a little hard to predict in terms of their approvals in this setting, but I do think they have a very good chance of being approved in late stage colorectal cancer. And I'm likely going to use them before I use regorafenib. I don't really use regorafenib in this setting, truth be told, because I don't think there's a lot of juice for the squeeze. I think there's a tremendous amount of risk in that drug. I've had patients not do well on that drug, and the median PFS benefit you get is less than a month. The overall survival benefit is roughly a month. It's just not good enough from my perspective. Lonsurf, I actually feel the same way about. So this drug is nice because it's a VEGFR 1, 2, 3 specific inhibitor. It doesn't have a lot of off-target tyrosine kinase receptor effects. So I think you're going to hear a lot about this drug. So please keep that in mind. Tazemetostat, we all know, it's an EZH2 inhibitor approved for epithelial sarcoma with patients who have the appropriate EZH2 mutation, and also approved in follicular lymphoma, actually irrespective of EZH2 mutations. It's a decent drug. I think the company thought they'd make a lot more money off of this drug. I just read an article recently saying that they were a little surprised they'd make as much money as they thought they would. Then there's this other drug, it's a Syk kinase inhibitor. It's called sovleplenib. Now, the only Syk kinase inhibitor I know that's approved right now is fostamatinib. We use that in ITP, right? We use it pretty late in ITP. So you can see that they're trying to test this in ITP, a lot of the lymphomas. In ITP, I think even if it's got a positive result, it's going to be very hard to find a footing there because we have a lot of the thrombopoietin analogs, and we've got, avatrombopag, lucitrombopag, lucitrombopag, and eltrombopag. We also ramiplostim. We've got, you can do a splenectomy, you can do Rituxan, you can do CellCept up front. So there's a lot we can do in ITP now. So this is a late stage sort of drug for ITP. So I don't know what sort of footing it'll find there. Then they've got a PI3 kinase delta inhibitor. That's in a lot of the lymphomas. That's also a little bit of a challenge. You can see that a lot of the PI3 kinase delta inhibitors in lymphoma, particularly in follicular lymphoma, have had a rough go of it. So umbralisib was taken off the market, duvelisib taken off the market despite getting initial approval. Idelalisib, which was approved in CLL, was taken off the market because of toxicity, a lot of GI perforation. So when people inhibit PI3 kinase delta, there's a challenge there. So I get nervous about it. I actually did an entire interview for Targeted Oncology on this. There are some concerns here, but interesting that they're going after this. They have some other drugs, FGFR 1, 2, 3 inhibitor, IDH1, 2. So we know about the FGFR inhibitors. In fact, pemigatinib just got approved for FGFR1 alterations in myeloid and lymphoid neoplasms. That was last week. Pemigatinib, infigratinib, both FGFR2 inhibitors approved in cholangio. And then you've got erdafitinib, FGFR2, 3, fusions or alterations in bladder cell urothelial cancer cells or cancer tumors already approved also. So there's a lot of competition in this space. They have an IDH1/2 inhibitor, so that's interesting, right? Because right now we've got ivosidenib, which is an IDH1 inhibitor approved in AML. We have NSCLC, which is an IDH2 inhibitor approved in AML also. So it'll be nice to have a little competition here. It's nice to have new drugs, hopefully, that can enter this market. That's good. They have a MAP kinase pathway inhibitor, so really it inhibits ERK. They have a BTK inhibitor, a lot of competition there. It's really hard to get into the BTK field. You've got ibrutinib, acalabrutinib, zanubrutinib's, already approved essentially in CLL. It's going to be approved in CLL. It's a very good drug. It's in mantle cell. Zanubrutinib is excellent, and pirtobrutinib is also in trial right now. So I think there's a lot of really good drugs in trial. They have a CS1R inhibitors, a lot of different molecules that target CSF1R already, and they have a CD47 inhibitor. So I actually had very high hopes for CD47 antagonists, but what we're finding is they have a lot of on-target toxicity because they're CD47 receptors on the red blood cell. So margolimab, which is the Gilead drug that they basically purchased from 47, has been a challenge, right? They've had a lot of toxicity. It was a hold in phase 3, my MTS. And so there are some concerns there. In fact, so much so that AbbVie recently dropped their entire CD47 program. So whereas I had a lot of high hopes for CD47 inhibitors, now I'm very concerned about what's happening there. It's really interesting. So that's the Hutchmed pipeline. I think it's interesting. Ram, any thoughts on this?
Yes, as you could see, this is a lot of these drugs are basically from China. And some of them are approved there for treatment, savolitinib, as you can see, and I think that's a very robust pipeline. But at the same time, a lot of the other drugs, their VEGFR inhibitors and their CD47, I think really needs to prove their place in the field. And recently the FDA said, if all of your clinical trials are done outside of the United States, then approval may not be a guaranteed approval. And that actually hit that VEGF inhibitor that they were looking in the neuroendocrine space, actually. So there is some evolving changes in how FDA approves, evaluates and approves these cancer drugs. So we need to see, and that's going to be a lot of changes are going to be happening in that space, I think.
Yeah, I agree with you. I think the FDA has absolutely said you can't do China-only studies anymore. Several PD-1 inhibitors got hit by that, particularly in lung cancer, as you know, recently. So they had really good data, but it was China-only studies. They didn't get approved. Eli Lilly got hit with that pretty hard. So, I think that the FDA has kind of put their foot down there. You have to do a mixed study. You have to have some US patients. So, all right. So we did this poll. Essentially everyone feels like the pipeline's not too shabby. So I would agree. I think that's pretty reasonable. Ram, what do you think? How would you answer this?
I don't disagree. There are good drugs, a lot of science behind it, but nothing that you would say is this blockbuster that's going to make a change in the market immediately.
Awesome. All right, let's do case one, Ram.
So first case is a patient with ALK fusion positive non-small cell lung cancer. Now ALK fusion, this is like old news in some ways because we've known ALK fusion as physicians that take care of patients with lung cancer. ALK is a well-known driver target. We know to look for it. And there is so many ALK inhibitors now available that you are sometimes pressed for choosing which one is the right drug in a clinical situation. So why this case? And as we go forward, you will see why, because there are some interesting issues with ALK patients because we still don't cure these patients. We still have to deal with progression after several of these new ALK inhibitors, and then we struggle how to best take care of these patients. So this is a 66-year-old gentleman who presented with a left axillary mass, but on further imaging with a PET CT was found to have more diffuse disease, which is what you see on the PET scan. You can see the left lung mass, which is extending into the mediastinum. You have these supraclavicular nodes, and then also the axillary mass. So quite extensive disease. So biopsies were done, which proved this was an adenocarcinoma of pulmonary origin. And case in point here, PD-L1 expression high at 95%. A lot of these tumors have high PD-L1 expression, but wait for the molecular testing. You have a better target with ALK rather than giving them PD-L1. And we are seeing a lot of data that PD-L1 with chemotherapy or by itself may not be very effective in this population. So it pays to wait, get the testing done, and then proceeding on treatment. So this is the test report. And you can see that there is an ALK fusion. Actually, there are two of them. There is the well-known EML4 ALK, which is common. And then there is this ALK PPP1CB, non-canonical fusion. And we also see some other changes at PP53 mutation, but we also see amplification in MET and CCNE1. Now, the MET amplification we'll discuss down the line as to why that is important. But as we do better, as we get better and better in the next generation sequencing, it is not uncommon to find non-canonical fusions. They do happen. And the fusion partners you'll see can be of different varieties of genes. And particularly if you do RNA-seq, you tend to find more of this. But EML4ALK is the most common. That's what we see here in this pie diagram here. Those are quite common when it comes to EML4ALK. And you see these non-canonical fusions. You don't necessarily have to change your treatment approach just because you find a non-canonical fusion. ALK is still ALK, still actionable. So we saw ALK fusion, and you can see that that's what this report indicates here, both for the variant 2 EML4ALK as well as a non-canonical fusion. The indication for the drugs are the same. To summarize, therefore, this is an ALK fusion positive patient with MET amplification, low tumor mutation burden, microsatellite stable, and a very high PD-L1 expression at 95%. Go ahead.
Thank you so much, Ram, actually. So for this one, so as a variant scientist, what are the things we will gather from this kind of comprehensive genomic profile reports? So when we find these reports, first thing we will dissect gene by gene to understand the tumor biology. So straight away, we will not go for the treatment per se when we are saying ALK and all those things. So we need to understand the tumor biology, how much it's important, contributing to the tumor, as well as from the tumor progression perspective. So let's dissect one by one. So the first thing we need to look at the very much the driver mutation. So among the one Ram have presented, who is the driver mutation for this cancer most likely, right? So the first thing could be possibly the ALK EML4, which is very well known. It's a gene rearrangement between two genes. The first gene is EML4 and the second gene is ALK basically. So when ALK as well as the EML4, these two genes combining together, it can form different kind of variants basically, right? Each variants have their unique property to cause the cancer as well as to progress from the cancer, and treatment response also, it will be totally different basically. So what exactly happening in the ALK EML4 fusion is that if you look at two genes, EML4 as well as the ALK one, the red marks over here mentioned are the breaking points of EML4 gene. Okay, so those areas are the one who is going to get break and going to fuse with the ALK gene. So if you look at only one breaking point is available, and if you look at the right hand side over here, also indicating the same thing, the ALK one having every time the breaking point is A20. So A20 is the nothing but the exon 20 region. So around that 20 region only the every breaking point is happening. But if you look at the EML4, very interestingly, it will have different breaking point joining together. So that's what it's making totally different. Variant 1 will have totally different one breaking point with a fusion with ALK A20, and then variant 2, variant 3, and all those things will have differently. How it is going to impact the cancer driving as well as the treatment response is that mainly if you look at the EML4 structure, it is having TD region, it's having basic region, it having help domain, and also having tap domain. So majority of them are going to differ in the tape domain. So the yellow color mentioned over here is the tape domain. Okay, variant 1 and variant 2 will have tape mostly, and whereas variant 3 will not have the tape. So what will happen is that like when the tape is not available, it will become like a more like kind of stable protein. So if it is more stable inside the cell, what will happen that it will continuously activate the downstream signaling pathway. Whereas the variant 1 and variant 2 are unstable in nature, but it will be getting stabilized by the other molecule, which we will discuss in the next slide basically. Okay, so this is how it's going to get differ from variant 1, variant 2, and variant 3. Among this variant 1, variant 2, and variant 3, which are quite common in the lung cancer, like Ram mentioned in the earlier slide. So variant 1 and variant 2 having good prognosis. Whereas the variant 3 will have the poor prognosis because variant 3 tended to develop the brain metastatic more often, basically. Whereas variant 1 and variant 2 will not develop more brain metastatic, but we need to look at that what other options are available for that variant 1, variant 2 to cause the cancer and progression from the normal TKI treatment. Okay. So this is the effect of alectinib in the ALK EML4 variant. So basically, when the ALK EML4 fusion happening and ALK EML4 amplification or point mutation happening in this ALK EML4 gene, what will happen is that it is going to activate the downstream signaling pathway in various different ways, basically. So it can activate PI3K AKT signaling pathway, or it can activate the JAK signaling pathway, or it can activate PL3, and also it can activate RAS-RAF signaling pathway. So what we need to do, like clinicians like Ram and Basim mentioned, that the big headache will be what will be the right TKI to select, basically. So which means that we need to make sure that the TKI we are selecting, it can able to control all the signaling pathway which can possibly come out of this ALK EML4 fusion, which is all these 4 signaling pathway I'm talking about. Okay, so if you look at the alectinib, and the right hand side, the B graph, if you look at that, the phosphorylation of the scat molecule, the variant 1, variant 2, variant 3, right? So minus and positive, which means that without alectinib and with alectinib, plus means with alectinib. So whenever the alectinib was given to the patient, you can see here the phosphorylated form of the scat molecule is going down, as well as the phosphorylated form of the phosphorylated form of the ERK molecule is also going down, and also the phosphorylated form of the AKT molecule is also going down, basically, especially in the variant 2, not variant 1 and variant 3, especially in the variant 2, when alectinib was given, all this signaling pathway can able to control, basically. This is what we wanted, right? So we need to make sure that the entire pathway can be shut down when we are selecting the TKI particularly. So that is the best thing we need to do, it to control the entire downstream signaling pathway to move further, basically, right? So in this case, we are discussing about variant 2 specifically. So which means that when we are selecting among the TKI, alectinib, there is a high chances that you can control all the signaling pathway. Whereas when you are selecting other TKI, we don't know whether it can able to control all this downstream signaling pathway. Another perspective of that is that when you are selecting crizotinib, that kind of TKI, what will happen is that it can able to generate the variant 3 down the time and then causes the brain metastatic. That's very common. But crizotinib also having another property to control the MET inhibitor. So it's a point of debate that which TKI to select among these ones still now. So again, as I mentioned, that the variant 1 and variant 2 are quite unstable. So who is the molecule inside the cells going to give the stability for these 2 variant is HSP, heat shock protein 90. So the heat shock protein, if you look at that one, it's in the yellow color one. So it is going to stabilize this variant 1 and variant 2 to keep them quite active state. So that's why you are going to see that a lot of HSP90 inhibitor can also possibly combination treatment for this TKI in order to control the variant 1 and variant 2 actually. So you will see in the future that quite combination of treatment when variant 1 and variant 2 of ALK inhibition you are selecting, you can give the combination with the HSP90 also. Why I'm mentioning is that if you look at the right hand image, that variant 1, variant 2, variant 3, when it was treated with the HSP90 inhibitor, so you can see the downstream of the variant 2 generation. So you can see variant 1 is also getting downstream and then variant 2 is also getting downregulated. So that's a very clear indication that when you are combining this ALK inhibitor with this HSP90, it will be a fantastic molecule. But again, the dosage correction will be the things we need to do. Maybe clinicians will take the best decision in the future. So this kind of clinical trial may also come in the future, basically. So these are some of the clinical trial which is going around. So, but if you look at that, most of them are in the phase 1 and then some of them are in the phase 2, and then only very few molecules are comes across after the phase 2 actually, right? So still it is a long way to go. It is a long molecule which is studied for so long period, but still it is not yet completely explored. So still we need to go through that one.
Thank you, Salambalas.
I will hand over to Ram.
Yeah. So in terms of how we approach treatment for patients, as I mentioned earlier, there are several ALK inhibitors And as you can see, if you look at the number of ALK inhibitors that I put down here, there is at least 6 of them that's approved and available. Some of them, like savolitinib and crizotinib, are the first generation inhibitors. And the field has moved towards the second generation. And we were talking about alectinib, and Silambarasan was discussing that. And we also have brigatinib and ensartinib. Now, lorlatinib that's here is actually, I put it there, but it's actually a third generation ALK inhibitor, and we'll touch on that. And the reason that we go with the second generation ALK inhibitors is that they are better drugs. They've been designed to address some of the limitations of the first generations, which is limited CNS penetrance. They are more promiscuous across kinase targets, but these are very specific. These second generation are specific to ALK inhibitors, to ALK kinase. And therefore, the toxicity management is also better because they don't have widespread non-ALK kinase activity, which can lead to toxicity. So these are some of the first choices when we decide to treat these patients, particularly alectinib and brigatinib are kind of the first choices to choose from because we also have a lot of experience. So what happens if we have some difficulty after somebody is on treatment with second generation, either because of toxicity or progression? Then we can potentially cycle through. And so there have been studies that have looked at patients who have been on second generation ALK inhibitors. And then after progression, they were tried on different drugs. And the most data that we have is with savolitinib, brigatinib, and lorlatinib. So in majority of these studies that I've listed here, which is phase 2, most of them, and the ALTA-2 trial, were all patients primarily on treatment with alectinib in the front line and progression. And then subsequently, they went on to get one of these drugs. And as you can see, the lorlatinib phase 2 trial actually was quite robust with 139 patients. Similarly, brigatinib with the ALTA-2 was 103 patients, but we don't have a lot of data from that yet. But you can see that the response rate for lorlatinib in patients who had failed on alectinib or other first generation ALK inhibitors was quite good at 41% in the second line setting. And the median progression-free survival is about 7 months. Again, these are robust second line options too in the event of progression after a frontline treatment of the second generation tyrosine kinase inhibitor. What do we do after we've given up on all of these ALK inhibitors? We've tried them all. They're not working anymore. And that's when we come back to our trusty old chemotherapy regimens. And the to-go regimen used to be a platinum plus pemetrexed or a platinum plus taxane, but the IMpower 150 study looked at patients with both EGF. It was actually designed for all comers, where the patients received carboplatin, Taxol, bevacizumab versus the same regimen plus adalimumab, which is a PD-L1 inhibitor. And what we found was that it was effective, but more specifically the EGFR and ALK population, addition of adalimumab did improve, which is unique in the sense because all the other studies that have looked at PD-1 and PD-L1 inhibitors for patients with ALK inhibitor, there wasn't efficacy. In fact, in some of these patients, there were some significant adverse effects and worse outcomes. But the addition of a VEGF inhibitor along with a platinum doublet seems to show a better progression-free survival. And it was, the hazards ratio was also quite significant there. Of course, this is a sub-analysis. The original study was not powered to look at this population. So something to keep in mind when we run out of TKIs for these patients and we're looking at chemotherapy choices. So what happened to this patient? So initially diagnosed in August 2020, and they got started on brigatinib. And you can see the second column, tremendous response, complete response. All of that tumor activity that we had seen and the tumors completely disappear here, indicating an excellent response to treatment. But unfortunately, it was relatively short-lived here because as you can see, in a couple of years, in a year and a half, actually, you see progression here. And you can see it on the PET scan. You can see these lymph node activity in the mediastinum, which is quite significant. And so patient went on to lorlatinib, which is a third generation ALK TKI. And unfortunately, again, within a couple of months, this patient has progression. And you can see this activity here in this lymph node, and they switched to carboplatin, pemetrexed, and pembro. Again, interestingly, they have a tremendous response here, complete remission. But within like around 5 months, you have further progression. So case in point, choose your best treatment for the first line, because that's where you get the best effect. So you can see a 1.5-year disease control. But subsequently, you have responses, but they are relatively short-lived, and you have to sequence through the available options here. So we have a poll question here. Would you have used lorlatinib or started chemotherapy after this patient progressed on brigatinib?
Let me launch this poll really quick. Sorry about that. All right, let's see what people think. So this is my patient. So everyone agreed with me using lorlatinib, which is always nice. That's great.
Yes, that's validation. Agreed. So, no, I agree with that. I think lorlatinib is a very appropriate option. So
I will just say, Ram, really quick, if you just go back 2 slides super fast, just to clarify, just super fast. So this patient progressed, when they progressed in January 2022, they only had this lymph node here. So I had a lot of room for error, right? So it made perfect sense to just try lorlatinib here because there was so much room for error. You could have done it regardless, but there was a lot of room for error here. And clearly I lost yardage, so to speak. And so, you know, we had to switch here, but yeah, that's it.
That actually beautifully segues into what I wanted to talk about, which is the operational option. So for ALK fusion positive patients, when they have progression, this is actually now part of the NCCN guidelines too, but very recent update. You have to decide whether they have florid widespread progression or it's more of an oligoprogression locally. And if it is an oligoprogression relatively local, then an operational option or a targeted therapy, which is non-medical, you know, surgery or radiation, could be very appropriate, and you could potentially control the ALK inhibitor that they've previously been on, and that can give them a little bit more mileage to treat that focus of disease with SBRT, and then keep the, you know, the first line or the second line ALK inhibitor on board and try to get as much mileage as possible on these patients is one definite option for them. But then you end up with all of these treatments, and you've tried all of this, and they have florid progression across the system. What do you do? And that's where your subsequent molecular-based therapies, clinical trials become very pertinent and important. And to figure that out, next generation testing is extremely key, because we need to identify why they are progressing. So what is the resistance mechanism to the ALK fusion, ALK TKIs here, and whether they are on or off target mechanisms is what we need to understand. And that can give us an idea on how to treat the patient. So this slide here references some of the ALK resistance mechanisms. But just to summarize this, you can either have on-target resistance mechanisms, which is here in this left panel here, which details these ALK missense mutations like this I117X and then the F1174X. These are all mutations that happen in the ALK gene, which makes them resistant to the ALK inhibitor that we use. And this is more common when we used some of the earlier generation inhibitors like crizotinib. But with the newer generations that are coming, we are also seeing off-target mechanisms, which can be involving NRG1, EGF, a variety of these kinase inhibitors. And you can also have a little bit more rare mechanisms such as EMT, as well as small cell transformation. This is rare, more well described in the EGFR mutant population, but it's also now being seen in the ALK population. And what I've tried to do is kind of put the incidence of these different resistance mechanisms And as you can see, the ALK, the MET amplification-driven resistance mechanism is relatively common, 12 to 22%. And one of the things we believe nowadays is that these resistance mechanisms don't necessarily happen later on to therapy, that they actually exist. As in this case, in this particular patient, we did have MET amplification at the time of diagnosis. And it's a population selection because our drugs are so good, they knock out all these ALK fusion-driven cancer cells. The subclones that have the MET amplification then get selected and then tend to survive, and they then drive the resistance mechanism. Now, of course, we need to do subsequent sequencing, a lot of elegant next generation sequence studies to prove this, but it's a reasonable hypothesis in this patient's case, is how I would put it. Can it be that there are other resistance mechanisms at the time when they progressed? Since we don't have subsequent testing, we don't know for sure, but it's a reasonable hypothesis to proceed with when you're looking for options after they fail, usually the standard lines of care. So a quick segue into the rare ALK fusion partners from silambarasan, and then we'll get back to the case.
Yeah. Thank you, Ram, actually. So like this case is a very classical example that why we need to understand the tumor of entire thing, not just so in this case, we have started with one ALK inhibition, but also that there are other clonal populations also could be possibly present. So that's why we may need to take the approaches of MGTO based treatment approach, that maybe we need to give the combination of treatment in the early setting to control the subclonal population also in order to control the progression basically. So right now, earlier settings, we have used ALK EML4 fusion inhibitor and ALK TK inhibitor basically, but if you notice the CGP report that there is also one more fusion, which is the rare fusion, ALK PPP1CB mutations also present. So these are some of the fusion partner for ALK, which are rare in condition in a different cancer settings basically. So if you look at the lung adenocarcinoma, VIT, and all those things are coming up. And also very interestingly, in this case, what we are seeing is the PPP1CB ALK fusion, which is quite observed only in the glioma. So isn't it telling some other story that this clonal population have the potential to drive the cancer towards a brain metastatic? We don't know. It could be possibly only the patient tell the story that when they are going for the further treatment. So this PP1 fusion, how it is generated is that in between the exon 1 to 5 and the ALK as usual before the A20. So both of them are combining together to form the new protein. So new protein means this fusion itself could possibly have the potentially totally different property altogether. So it could also generate as a future subclonal population to drive the cancer further to escape from the current TKI, which may or may not work for the patient basically.
Thank you. So once we have identified the resistance mechanisms and we have a reasonable assumption as to MET amplification could be the reason here, what are our options? Now, surprisingly, when we research some of these clinical trials that are currently available, specifically to MET amplification, there is not a whole lot. And there are some potential drugs out there that we could explore, but a lot of us sometimes go back to crizotinib. As you know, crizotinib was originally developed as a MET inhibitor. And when ALK fusion was detected, then Pfizer quickly moved crizotinib to this population. And with Dr. Shah's leadership, they did this study where they showed the benefit for crizotinib because it did have ALK activity as well. And in this particular situation, crizotinib could be an option because it has the dual activity against both ALK and MET. So sometimes when we have a second off-target resistance mechanism, we may have to keep the original tyrosine kinase inhibitor, which may be the ALK tyrosine kinase inhibitor. And whatever the off-target mechanism is, we may need to target that with another drug. So you'll have to do a combination of two drugs, which will be difficult to do because of toxicity, and we don't have enough data on how they would work together. Here, maybe crizotinib is kind of nicely fitting into that scenario. And we talked about some of the ALK mutations that can also happen, which is an on-target resistance mechanism. particularly with these new second-generation drugs, we don't see a single ALK mutation. We end up seeing these compound ALK mutations, which makes it extremely difficult to target. Glittertinib seems to have some activity. As you know, glittertinib is a drug, tyrosine kinase inhibitor, that's approved in the use of leukemia patients. But there is some case reports, of course, case reports, not clinical trials, which are showing promise there. Now, what about the trial options? Now, I've listed a couple of drugs, TPX-01313 and NVL-655. Both of these drugs are fourth-generation ALK TKIs. And my apologies, I agree, that's a typo there for glittertinib. But these two drugs that are listed on top of the trials options are fourth generation. And as I said, with the first generation, they were promiscuous kinase inhibitors, not just to ALK. Second generation, they are highly specific to ALK. Third generation, lorlatinib, was designed simply because with these highly specific ALK inhibitors, it's a function of evolution, you develop resistance mutations. They saw a smaller macrocyclic molecule like lorlatinib can be effective against ALK and also reduce the chance of developing on-target ALK mutation type resistances. And that's the same principle that these two top fourth generation ALK inhibitors are following. Repotrectinib also has some activity there, but it's more driven towards ROS. And then there are some newer technologies with PROTAC, which is a proteolysis against protein chimeras and ALK neopeptides as well. But what about MET amplification? We can go into this. I know Bassam has a lot of discussion coming up with MET, so I'm going to move through this because it is a well-known. But what about the drugs out there? What do we know? On the left panel, what you're seeing is data from crizotinib. This was Dr. Kimmich's study. And what you see on the top panel, which says low, is low MET amplification patients receiving crizotinib, and their response rates, as you can see, is not great. And this was all done by fish-based MET amplification. And then the subsequent panel, the middle panel, is the sort of the mid-range amplification. Again, you see some activity, but the high amplification that you see down here, you can see some really good responders there. And on the right side panel, what you're seeing with capmatinib is the same thing, where patients with high amplification, where this was done with fish, that was done with next-gen sequencing, copy number of 10 or higher, you can see really good responses to, in this panel, on panel B, you can see the MET amplification, and you see a good response rate there, about 40% or so. And when you look at the progression-free survival, though, what's interesting is it was only about 4 months. So there is response, but you're not seeing a durable response for MET amplification. Again, these are not ALK positive patients. These are patients with only MET amplification without ALK. But this data shows that there is activity against MET amplification with this tyrosine kinase inhibitors. So definitely a consideration. We have another poll question here. What would you do in this patient who progressed after brigatinib, lorlatinib, and chemo plus PD-1 inhibition?
Just to clarify, that second option would be that you would continue your ALK inhibitor, so like lorlatinib in this case, and then add the MET inhibitor. So the question is, are you going to move to chemo, which would be taxotere-ramucirumab, that would be your next option? Are you going to just add the MET inhibitor to address that MET amplification? So hopefully you add capmatinib or tevotinib or whatever to your ALK inhibitor. And then the third one is, would you add back brigatinib to Alimta Keytruda and say, hey, you've already done your carbo Alimta Keytruda, you've done 6 cycles, you can't do carbo anymore. Would you do Alimta Keytruda maintenance and add back brigatinib? Those are the 3 options. Let's see what people have to say here. So, all right, go for it. Okay, so looks like about 2/3 of people would prefer to just add a MET inhibitor and stay on lorlatinib. One person wanted to go to brigatinib in the context of Alimta Keytruda. So, all right.
Gives sense. I agree with that. And so going forward, some of the trials we'd like to see, this trial, which is ongoing, looks at lorlatinib in combination with other TKIs, including MET amplification. So that's one of the trials that is available out there. And as I said, for the MET activated lung cancer, including ALK patients, crizotinib is a consideration for sure. And in terms of how we will be seeing the use of ALK TKIs, this is the current functional view, first generation, second generation, and third generation. But once TPX and so the NUV655 become available, we may end up targeting not just a single mutant active ALK TKIs. I think our shift will be from the generations to what type of ALK resistance mutations we're going to run into. Is it going to be a single mutant or is it going to be a compound mutant? And we're going to distribute drugs like that. And when you have a different off-target mechanism, maybe crizotinib will be there. And one of the other interesting things that may need to be explored, something that I would like to see, is look at ADCs in this population. Like we know telisotuzumab vedotin that targets MET protein as an ADC, maybe in combination with ALK TKIs, could be an option here. So I'm going to skip the slide and get going with the case 2 with Basim.
Yeah, I think what we'll do today, just because in the interest of time, this case is going to take a while. So we'll probably save this for next week. There is a question, Ram, that I'd like to have you answer. It's basically a question from Christian. Christian, do you want to just come off mute and ask the question or talk about what you want to talk about here? Dr. Christian, it's Cranton, sorry, Dr. Cranton Bose.
Hey, hi, Basim. Thank you for letting me in. And yeah, my question is pretty simple. That when you have MET amplifications along with ALK fusions, how are you looking at basically adding the downstream MET inhibitors along with the ALK inhibitors, and what is the comparative rationale? So yeah, thank you.
Yeah, thank you.
Absolutely. No, that's a great question. See, right now, the combination of combining a second generation MET inhibitor with another tyrosine kinase inhibitor has not been well studied now. But we know in the EGFR space that it's doable. We've done it, and it is active in EGFR mutant population, that when you have MET amplification as a resistance, you could do that. So I think more studies are needed in the ALK population. So in my practice, after like what Basim has gone through, would I add capmatinib on top of brigatinib or alectinib? I'd be a bit careful off clinical trial, because 2 tyrosine kinase inhibitors toxicities, I'd be a little bit concerned. But if there are no standard of care options, we could try it. But in this case, I think trying crizotinib downstream after you fail the second and third generation is a reasonable option, because it's approved in this population. We know its toxicity better, and we know that it has pretty decent activity against MET amplification, like we saw, particularly when it is significantly amplified. So that is probably one of my choices in this patient, either on or off a clinical trial. If I could give them crizotinib, that's where I would probably go in this patient.
Thank you. And, this may be a bit off track, but do you have any experience with the recently available chemo sensitivity tests and kind of personalizing the cytotoxic chemo drugs? Because that is another potential option in these multi-line failed patients if we were to, kind of add crizotinib with a non-platinum, non-taxane drug option, because, we really know that maybe those weren't working. So,
right. Yeah, there are some panels out there that are commercially available, but the data behind them is somewhat limited. But I think with the ADCs coming around, I think, can you do it? Yes. The short answer is that. But is it great data? Not really at this point. But as ADCs come on board, we may have better knowledge on how to choose our chemotherapy options. And I'm sorry, I need to jump off this meeting. But
yeah, no problem. Thank you so much. Yeah. No, Sharantan, how do I pronounce your first name? How do I pronounce your?
You're absolutely correct, Basim. It's Chirantan.
Chirantan. Okay, excellent. So yeah, there's a lot of people in that space, as you know. Cayutech, there's Cirrus has this. They do a lot of where they take patients' tumors, they inject them into mice, and they subject them to different sorts of treatments, as you're alluding to. There's not a lot of data at the moment to support the use of those studies in guiding your treatment. They've been around for a long time. People talk about them like they're new, and they're not, right? They've been around for decades, where people have done this and made money basically telling patients, hey, we do this and it works. And I've had recent patients consult me with this information in hand, right? They've done it on their own and they come to me with this data and we have to incorporate it. We don't really know what to do with it, right? I think in late stage situations like this, there's no harm in it. I think that's what you're alluding to, Sharon. Why not do it, right? It's okay to do it. If you got nothing else you want to throw a Hail Mary past, but then you say to yourself, what do I do with this information? Is it real? Like, let's say I take a patient's tumor and I inject it in a mouse. Well, does that really emulate the proper tumor microenvironment that you would see in a human? Is that really what we're actually going to be able to use? So what people are doing now, as you know, is they're moving on from mouse models and now they're doing it, in tissue culture and really doing like organoids. And they're saying, okay, well, we can emulate the actual patient's tissue, right, in a human model, and we can subject the tissue without having to kill a bunch of mice, right, which you never want to have to do if you don't have to. They're saying we can take these organoids, we can subject them to the various drugs in a tissue-specific manner and then use that information. So those studies are ongoing. It'll be interesting to see what happens with those. I mean, my feeling, Shranton, to be honest, is when you start to get to the bottom of the barrel, you can do whatever you want. I'm never going to say you're wrong, right? You can try it. But in this case, I wouldn't know what to do with the information. There's just no randomized control study, no data to really support you. And I think that's why those companies have had such a hard time getting off the ground, right? I think that's why, because there's not a lot of data.
No, I really appreciate your very candid and open response to that. I'll be happy to share some of our in-human in vitro organoid circulating tumor cell chemosensitivity data with you offline. So thank you. I didn't want to distract from the course of discussion, but thank you.
No, I think that's great. I think the organoid, like I said, that there's potentially a future for that. So I'd love to see that data. It's excellent. Thank you so much for that. So, this case, I did this case. It's actually pretty impressive. We're going to do it in 2 weeks from now. I don't want to keep people, I don't want to go over an hour. This case is exquisite. We also have an MSI high prostate cancer case that is mind-blowing. We'll do that in 2 weeks also, but we will really focus on MET exon 14 skipping in 2 weeks. I think in terms of concluding remarks, please know that you need to resequence at progression when you have a patient with an ALK fusion. You absolutely have to do that. You have to look for MET amplification. You need to switch drugs. You can try using crizotinib in this case. You can try adding a MET inhibitor. Ram is a little bit more cautious than me. In my career, I have no issue trying 2 drugs if I can get them, from the FDA and seeing what happens. As long as I know the safety profile of both, I don't mind seeing what happens in a case where you're desperate. In this case, what I will say, which is interesting, is one thing about this case, and it kind of gets to an article I wrote for biopharmatrend.com in my editorial series, where I wrote an article about quarterbacking a patient's cancer care and really talked about how you have to know a patient's specific situation. So if you look at this patient, for example, right, if you see where they're progressing, so we started them on brigatinib, they had a complete remission. We switched them to lorlatinib at first recurrence. We did do another sequencing, by the way, we did biopsy here, and it did show the same fusions as the initial situation. That MET amplification was there before and after. That was not new, but I wanted to make it very clear that we did do resequencing. I always resequence at progression. So we did resequence, and because there was such minimal disease here, I felt like there was certainly no harm in doing lorlatinib, like everyone agreed with, and that's where we were. We did chemo immunotherapy. Now, a lot of people in patients who have ALK fusions will say immunotherapy doesn't work, right? This patient had a PL1 that was 95%. And so on the off chance that Keytruda would benefit them, I did include it here. I would never do Keytruda alone, right? That's not a patient you want to do Keytruda alone because they have an ALK fusion. You typically think that immunotherapy alone will not work, but if there was any incremental benefit we could get from that, we tried it with the idea that we would try and go to Alimta Keytruda maintenance on the back end, and then maybe we'd see where we were at that point, Alimta alone, Keytruda alone, whatever. But bottom line is they did exceedingly well, right? They had a complete remission here, and then had a recurrence, very minimal recurrence in these lymph nodes. So what I'm alluding to here is if you look at where they recurred, right, in these 2 regions here, they did not have this disease while on the ALK inhibitors. So on the ALK inhibitor, that's not where their recurrence was. So you already know, right, that there's heterogeneity in this tumor. You can see that because these lesions at the beginning have not come back, right? So you know that different areas of this tumor clearly have different profiles. There's something about them that's different. So the thought you have is you say, well, wait a second, this is a very minimal amount of disease, right? There's very little here. Could I just add back the brigatinib, which was controlling that region, right? Could I add back the brigatinib, stay on Alimta alone, right? Because it's controlling everything else, and see what happens there. So that is an option. That's why we talked about it. But that's kind of why I think you absolutely have to look at scans. A lot of my colleagues don't, but you really need to look at them, and you need to look at them side by side, because you need to say, okay, there's tumor heterogeneity here. What was this responding to? Did it actually relapse here on the ALK fusion inhibitor? And that's actually not the case. So it suggests that maybe the ALK fusion inhibitor does control this region, right? But you lose ground elsewhere. So maybe you need to combine these drugs. And that's actually what I'm doing here, because there's such a minimal amount of disease here, as opposed to going straight to my MET/ALK inhibitor. What I did, actually, to be honest, is I started back brigatinib and I kept my Alimta, right? So this patient's on brigatinib Alimta, and I'm going to rescan in 2 months just to not lose any ground. I'll go immediately to, crizotinib after that. But that's what I did here. So, very interesting case, but this is why I think you need to look at the tumor from start all the way through progression and find out exactly where you are and to define that tumor heterogeneity. So we're going to stop here. I think that I sincerely appreciate everyone being here. Does anyone have any questions or comments before we go?
I mean, great case and great review, Basim and Dr. Ram. Thank you for this. You may want to consider an adjuvant radiation setting in this patient because the disease is so localized. I mean, that's just a comment. I think it was discussed earlier, but it's going to be worthwhile, I think. Yeah.
No, 100%. So that's the next step, right, Sharon? And the next step, so I typically want to get a feel for the tumor biology before I do that, right? So if the next scan looks like this, we will just radiate those lesions and be done with it. But there's actually 3 here. It doesn't look like there's 3, but there's actually 3 that popped up all of a sudden. So I'm trying to get a feel for the tumor biology, and the next scan, if it looks good, we'll do that 100%. So I'm very aggressive about radiating all of the metastatic disease. So completely agree. Excellent suggestion. Any other thoughts? All right, wonderful. Thank you for coming. We'll be back in 2 weeks. We'll do MET exon 14 skipping, and we'll do this incredible MSI high prostate cancer case. Thank you everyone for your attention and time. Please have a wonderful weekend.